Good evening, it's Doc Kyle Solano, and tonight we're gonna talk about enzymes, specifically enzyme kinetics. So, so what are enzymes? No, enzymes are proteins, and they are what we call catalysts. Catalysts increase the rate of the chemical reaction. So, why do we need to increase the chemical reaction? In the first place, spontaneous chemical reactions do tend to occur, but they tend to be very slow about it. So there are four ways to increase your chemical reaction. Number one is to increase the reactants concentration. Number two, we can have increased heat. Number three, we can have mechanical agitation. And number four, we can have the use of catalysts. Example is your coffee. If you add coffee granules and you add sugar granules, butangan mo tubig na dili init, eventually you're gonna have your coffee. Eventually, no? Maybe mga tulo ka birthday nimo, you will have your optimal blend of coffee. So, but if you add heat, then you will have your optimal blend faster, especially pag you add mechanical agitation. Especially kutaw na nimo na siya. What happens in that solution, di ay? Remember that it's still a solution. Your solutes are your coffee granules and sugar granules, and your solvent is water, hot water, in fact. So, what happens? In the solution, you have your atoms of your coffee particles, and I know. So, let's take another example lang. So, we have two chemicals, you have two molecules. When they bind together, there will be a chemical reaction. Remember, in the context of your chemical reaction, the solution, the solvent is very big compared to the size of the atoms. That's why they will just lutaw-lutaw around the solvent. Eventually, they will bangga and then there will have a chemical reaction. If you will increase heat, there will be an increase in kinetic energy. There will be an increase in the agitation or the movement or vibration of your atoms of your reactants. That's why makusog sila lihok. And mas kusog sila lihok, there are more chances of them hitting each other and creating a chemical reaction. Same if in that solution you had mechanical agitation, mas dako ang ilang chance that they will encounter one another. Your catalysts, they are in the reaction with your reactants, but what they do is they bind to the reactants together, and then they will. Remember that your reactants are 3D. They have a 3D structure, and not all of their sides are optimal to have a chemical reaction. So your your catalyst binds them in a way that both of their optimal sides face one another to produce a chemical reaction. So cutting that time in half na nagalutaw-lutaw lang sila sa solution. So apparently it increases that the reaction rate up to 10 times or more, no? So in your enzyme. So that's how important this is. So it reduces the energy of going around looking for another reactant to bind with. It reduces the energy and the time of it a lot. So that's your catalyst. Enzymes are protein catalysts, meaning it is produced by any biological creature. It can be produced by your bacteria. It can be produced by your plants and your animals. Just so happens that your viruses don't produce it because your viruses are just information. It's just data. Data lang dyan ng viruses nimo. The context of data transcribed in your RNA or DNA. They have enzymes in their capsules, but they don't produce it. They infect other cells for it to be produced. So going back, all the enzymes I'm gonna talk about today are in the human body na context, dili plant or animal. So we're gonna talk about these enzymes. They were discovered sa una when people were making bread. They add yeast, and then the yeast had these enzymes that will produce gas so that the bread will rise. no? It will will increase the reaction. Parehaning Goku, Super Saiyan. Wah! No? So ano mahitabo sa mga chemical reaction pag na-enzyme? Kusog na kayo siya, and it's very fast. So it increases the rate of the chemical reaction exponentially by 10. But it is bound by the limitations of proteins. Remember that your proteins are they're quite resilient, but they have lots of factors that may inhibit them, which we will talk about later. So thermodynamic spontaneity cannot tell us whether a reaction will be fast. So, like I said earlier in the example I said, uban na reaction, pag ihalo, you don't even need to add hot water or make mechanical agitation. The reaction goes fast. But there are some times that are slow. No? So we don't know that. So the speed of the reaction is a kinetic property controlled by the nature of the energy state of the enzyme-substrate complex and the transition state. So what is this enzyme-substrate complex? So your enzyme is this one. This is the active site. And your substrate is the reactant that goes into the active site. This is your active site. And once it goes into the active site, the chemical reaction now proceeds. No? Siya kadali. So substrate is the one that binds to the enzyme, no? Or the enzyme binds to it. It's the target of the enzyme. So it can be a lot. Substrates can be a lot. Nucleic acid, carbohydrates, lipids, proteins, and other enzymes pa usahay. Una yun niya. So kato, you can see here Finn is acting like an enzyme. No? So this is Mr. Fox. I forgot the name of this duck. No? Kanang ginabuhat sa enzyme. They're facing the most optimal parts of the reactants each other so that the chemical reaction will rapidly progress. Same with here. So they are creating a situation where the distance between the transition state and the ES complex of an energy diagram is reduced. We're gonna talk about that later pa. Like next pa siguro. Chemical reaction may go faster at higher temperatures. Like I said earlier, if you increase the temperature, there will be increased energy. No? There will be increased kinetic energy. Kay, I want you to, kung nana kay enzymes, giadan pa dyan nimo og heat. So remember that your enzyme is also a molecule. It's also affected by heat. So it rapidly moves around the solution pod, thereby increasing its chances that it will bind to a substrate. But if like I said earlier, your enzymes is a protein, and proteins are prone to denaturation. So I read before that per 1 degree in the body, per 1 degree increase of the temperature, there will be a subsequent 10 times increase in your enzymatic activity. Eventually, it will get too hot, it will denature itself, leading to a 0 in enzymatic activity. Eventually, mag-coagulate, mag-denature lang dyan na imuhang mga enzymes. No? As we all know, denaturation occurs when there are external factors like your heat, tapos ay strong base, strong acid. Same with your egg, pag lutuon mo na denature na iyang ang albumin, so nahimo na siyang lami na itlog. No? So kato. So but before mag-denature siya, completely denature, nag-increase din yung reaction na. I want you to understand that. No? Oh, sorry, na siya. Actually, so enzymes and substrates are attached to each other via non-covalent interactions. Why is it important for it to be a non-covalent interaction? Because once your substrate is converted to your product of your chemical reaction, it still needs to dissociate from your enzyme. Katano? Ano siya sa yun? That's why dapat dili siya covalent. Dapat dili siya permanent na bond. So the active site is specific amino acids in specific orientation where it can bind the substrate. Like I said earlier, we have two models of binding. You have your number one, your lock and key, meaning your substrate will bind perfectly to your enzyme, meaning your enzyme has a rigid active site, meaning it will not change. So if meant to be sila, meant to be dyan sila. And then we have another model which we call your induced fit, meaning your enzymes your enzyme's active site can bind, I can accommodate or can conform pa. So what is this model? No? Just simply means we really don't know what happens in the body. As you progress further in your study of biology, kay biology magyapon itong ginastudihan sa medtech, no? And in medicine, you will understand that there are a lot of things that we still don't know. We have a good idea on, and one of this is how substrates bind to enzymes. So does it matter? Yes, it matters because there are some substrates. Because there are enzymes that can bind different types of substrates, different types of isomers of substrates na may mga minute variations. So, more or less, kung lock and key jud siya, rigid jud kay siya, dili jud siya dapat it shouldn't be able to bind the isomers, no, or the other forms of the isoforms rather of the substrate. But since it can still bind and it can still produce a product, then that's why they thought of this induced fit. There might be some accommodation happening in the active site. No? So we're gonna talk about the kinetics of the enzymatic reactions na. So remember that in this graph is it's a beautiful explanation. So this is your substrate concentration. It's going up, no? And then this is your velocity. What does this velocity mean? Velocity is the rate of production. Nana kay products ba? Produce. And then substrate concentration, it's So, first order kinetics simply means na there is a rapid increase in production of products during the initial increase of your substrate concentration. Infinity ni. So, rapid increase meaning exponential increase. Eventually magplateau na siya. Ngano siya magplateau? Because unlike your substrate concentration and your initial velocity rate, the body of a human can contain only so much enzymes, no? So this is the rate limiting your concentration of enzymes in the body. So meaning if your enzymes only have a certain degree of concentration, then malimit pod imuhang maximum velocity, which is your Vmax. So I'll clean up the So if you find here in your first order kinetics, the rate depends on the concentration of the substrate. So your first order kinetics is dependent on the fact that your enzymatic concentration is still greater than your substrate. Eventually, pag sige lang gyapon ang production sa substrate, eventually mas greater na ang substrate concentration to your enzyme. No? And that's your zero order kinetics na. Okay? Because we have what we call your saturation. Meaning tanang active site sa imuhang lawas, no? Tanang active site sa enzyme sa imuhang lawas kay puno na og kanang substrate. They are saturated na. So they are saturated na. So therefore, dili na siya maka-proceed. It can't produce any more product because it has reached its limit. So remember, your zero order kinetics is not dependent of concentration of substrate. It is dependent on the enzyme concentration. Okay? So, and then kaning maximum velocity based on your enzymes is what we call your Vmax, velocity max. So if it reached already its Vmax, it is now your zero order kinetics. So this is the basic enzyme reaction. So your enzyme plus substrate. K is just the rate rate constant. And if you see this sign, kaning arrow na ana, it is reversible. Meaning, pwede pa nimo siya mabaliktad. So, next is your enzyme-substrate complex. Pag nagdikit na sila together, you can still pull them apart. But, once the enzyme and the product is already, if the product is already produced, you can turn it back to substrate. You need another enzyme to produce it back to your substrate. No, that is no longer a product. Basta pag nakabuhat na kag-product, it's irreversible to go back. And then we have this, what we call your Michaelis-Menten equation. No? So your velocity is just kanang ang velocity na ginamini ani is your 1/2 your Vmax. So the equation states that Vmax times substrate, right, over Michaelis constant ng Km plus substrate. So dagan kay siya mga formula, but simply put, your Michaelis constant simply means it is the concentration of your substrate. It is the concentration of your substrate, no? It is the substrate concentration that will give you 1/2 Vmax, no? So it is just used to calculate for 1/2 of your Vmax. That is your Michaelis concentration. What is the purpose of this Michaelis-Menten equation, no? So una, when we didn't have enough, the progress in immunology wasn't that advanced pa, by which we can have targeted quantification of enzymes, proteins, or other stuff in the body. Remember that your antigen-antibody reaction is the most specific reaction in all of biology. So we can have targeted, we just use an antibody to bind to the enzymes of need. But before, we didn't have that, so we tried to calculate the concentration of enzymes, current concentration of enzymes using its substrate uptake. I know, so it's hago kayo. So when we use the Michaelis constant, meaning if you use the Michaelis, if we're gonna look for the Michaelis constant, meaning malaman po nato ato ang 1/2 Vmax, and if times 2 lang nato ng 1/2 Vmax nato, we can have your Vmax, which will eventually find out the enzyme concentration by derivation. So what is the purpose of looking for or quantification of your enzymes? Remember that you have a lot of enzymes in your body. Usually these enzymes increase if there is damage to the tissue that hold them. Example, if you have myocardial infarction or heart attack, there will be an increase in release of cardiac enzymes, namely your creatine kinase MB, CKMB. So if there are increases in your CKMB, then we can assume that most probably there is myocardial damage. So turnover number of moles of substrate that react per second per mole of enzyme. So with two substrates, you can envision. So in this case, we already have two substrates. So it's more gubot than one substrate. So we have different types, no? So we have your ordered mechanism. This is A, ordered. So meaning, order siya. So your enzyme binds to A first, then it becomes EA, then it binds to B again, and then becomes AEB. So if you're gonna look at the arrows, it's just irreversible sequence siya. That's why it's called ordered. Then we have your random. So A plus E, AE can bind to EB. So EB can bind to A directly to form AEB. So basta nakakita kag daghag mga reversible na mga arrow, that's already random, ha? And then your 3, we have your ping pong. So what is this ping pong? Ping pong So this is your ping pong mechanism. So ping pong simply means your E enzyme plus A will lead to EA and will lead to a modification of E. That's why there is a bug list on top. So the modified enzyme plus product, and the modified enzyme can bind to your B. Can't bind to B without forming your product. Without being modified. So after its reaction, after making its product of B, it now returns to its unmodified version. That's why it's called ping pong. Balik-balik siya from modified to unmodified. Then, kato. So, like I said earlier, diba, your Michaelis constant simply means your 1/2 of your Vmax. So if you know your Vmax, then you can most probably quantify your your enzyme concentration. So Vmax tells us how fast the enzyme can generate product under saturating substrate conditions. Lineweaver-Burke plot is a double reciprocal plot. It's a graphical method for analyzing kinetics of the enzyme. So first we're gonna talk about this. The Lineweaver-Burke plot is best used for detection of what type of inhibitor is present. An inhibitor simply means that it inhibits the action of the enzyme. We have reversible and then irreversible inhibitor. So we have different types of inhibition. We have competitive, meaning it binds to the active site, no? Binds to the active site of the enzyme. So dili na makadikit diha ang substrate. So to reverse this, we just need to increase the concentration of substrate so that there will be an increase in affinity. An increase in affinity of the substrate to the active site compared to the inhibitor. Next, we have your non-competitive inhibitor. Your non-competitive inhibitor binds to the allosteric site. Now, what is the allosteric site? Remember that your enzyme is a protein, and proteins have a complex 3D structure. So there is also another binding site. Allosteric site simply means a binding site that is not the active site. So if an inhibitor binds there in your allosteric site, remember your allosteric site can facilitate, there is kanang activator, the enzyme is dormant unless there is an activator that binds to its allosteric site. Then there are also what we call inhibitors. There are different types of functions na makita niyo sa mga allosteric site. So since we're talking about inhibitors, so an inhibitor binds to the allosteric site, and there will be conformational change in your enzyme active site. So dili na makabind diha ang substrate. And then we have your uncompetitive inhibition. So it will still, this inhibitor, uncompetitive, cannot bind to the enzyme directly. It still needs to wait until the substrate binds to the enzyme. And therefore it will now bind to the complex, the enzyme-substrate complex. That is the only way it will recognize the enzyme and the substrate, no? If they are bound na. So kato, this graph of your Lineweaver, go back. You have your competitive inhibition. The Km is increased and Vmax is unaffected, meaning, like I said earlier, you just need to increase your substrate concentration, and what your Km simply means, diba, is concentration of substrate needed to produce 1/2 Vmax. So, increase lang nimo imuhang. You just need to increase your your substrate. So, you need to increase your, if you increase your substrate kanang level to remove all the inhibitors, your Km is also increased, but your Vmax, your maximum rate, no, of product production is unaffected, because like I said earlier, your enzymes are still alive, no, they are still active. You just need to increase your substrate concentration so your enzymes are not damaged. Remember that Vmax is a property, is or slash dependent on your enzyme concentration. So you have your uncompetitive, no, the ES complex, no, your Km is reduced and your Vmax is reduced. You have a reduced Vmax because once nag-bind na magud sa ES complex ng uncompetitive nimo na inhibitor, it can no longer be used, no? So your total enzyme population or your concentration is now decreased. So your total kato decreased. So since ninahog pud imuhang nag-decrease pud imuhang enzyme complex na kay nag-decrease pud imuhang activity. So nag-decrease pud iyang maximum na saturation. Okay, so your Km is reduced also because like I said, since ninahog pud imong Vmax, the ceiling is lower. So you need less substrate, for you to reach your 1/2 Vmax, no? And then your non-competitive inhibition, so your Km is unaffected. So you can still reach your 1/2 Vmax, but your Vmax is reduced because na-reduce naman imong total enzymes. So you can still reach your Km, but ang total nimo dili na nimo makuha. So a suicide substrate is an irreversible inhibitor. It binds covalently to the enzyme. Suicide substrates are important. So, example, if there are lots of enzymes na makadaot to your body, sometimes the body does things that are detrimental to itself. So, sometimes you use suicide substrates as medication to stop those enzymes. No? So, thank you for listening. That concludes my lecture for tonight. And if you have any questions, feel free to PM me lang. Bye-bye!